By default, the Grameen search interface searches across all genomes. There are 150 in release 68 of Grameen plants. The gear icon at the right of the search status lets you select which genomes to search within. Let's choose sorghum, maize, rice, and Arabidopsis. It doesn't look like much has changed because this search is limited to curated genes. But when you expand the homology tab, the gene tree has been pruned to show only genes from these 4 genomes. It also removes empty spaces from the multiple sequence alignment. If we click here to search for orthologs, the results are now limited to these 4 genomes. Each gene from a reference genome in a pan-genome site has a link to that site. Let's click on sorghum base to see the MSD2 gene in the sorghum pan-genome context. The neighborhood conservation display mode can reveal local copy number variation among sorghum varieties. You may also see annotation artifacts. Here, the flashing nodes indicate split gene models. There are many blue genes in this region from the same family. You can highlight a gene family to help identify copy number variation. Another way to visualize copy number or presence-absence variation in a gene family is to search for all the genes in the tree and study the taxogenomic distribution of search results. You can see there are a few sorghum varieties that seem to be missing a gene toward the end of chromosome 1. We can select some of these alleles and search for their orthologs to confirm this. It looks like one of these genomes might have a structural variation that relocated the gene to the other end of the chromosome.